Hi everybody, I'm Dave Tabb, nice to meet you all. We'll be working together this week on a program that we hope is going to be sponsored by Diplomix. Diplomix is a program run by the Department of Science and Innovation. This year, Department of Science and Technology has been joined by Department of Higher Ed to make this DSI organization. And so there's a much stronger emphasis now that Diplomix take on an educational role, and this gives us the opportunity to teach much more broadly this material. I normally teach a fair bit over at the University of the Western Cape, and of course at my home campus, the Tigerberg campus of Stellenbosch University. But I'm really glad to get to talk to people even more broadly than that. One of the key messages that we want to convey with this bioinformatics training is that you can gain a very powerful, very marketable skill set to be ready to do graduate training or postdocing or whatever at great universities. You have passed a lot of tests to get to this point in your career, and I'm hopeful that the addition of bioinformatics training skills will get you to a place where you can go to even better opportunities in the future. Bioinformatics is probably one of the most mobile of skill sets that everyone in biology is now needs these skills. And it gives you a much better opportunity to move to better and better labs by developing these skills. So I'm glad that you're here today. I'm glad you made it a priority to be here. And through the course of this week, we're going to cover a lot of material that is very foundational in the space of bioinformatics. Okay. So we're going to be talking about some of the most basic of technologies associated with bioinformatics. There's a world of people out there who believe that bioinformatics is all about sequencing stuff and about alignment, sequence alignment stuff. So these are really foundational issues, and I think that most people will find that a lot of bioinformatics topics don't really come naturally if they don't have background in the concepts of DNA sequencing, sequence mapping, and sequence assembly. And all of that is what we're going to handle this morning. So we're going to start with a little overview of sequencing chemistry. How many folks here would feel comfortable right now explaining how massively parallel or next-gen sequencers work? A lot of people, frankly, in biology treat them as black boxes, but I hope by the end of today you're going to have a better sense of how the old school sequencers compare to these new ones that we use. You know, if you throw your samples at an Illumina, how do you get data out of that? We're going to be talking about base calling errors. Essentially, how do we transform the signals that come from the sequencing experiment into letters, and how much confidence can we place on the individual letters of a sequence? We're going to talk about mapping versus assembly, which is one of the key differences in how we deal with the data that come back from sequencers. And we'll talk about the Burrows-Wheeler algorithm and suffix arrays, kind of a complex topic, but one that's important to think about. And then we'll talk about assembly, which is in some ways a mind-blowingly difficult thing, but which is in fact something that we're capable of processing on a desktop computer. Along the way, we'll talk about a few file formats that are rather important that everyone should be familiar with. Those will be FASTQ, SAM and BAM, and FASTA. All right. I think everyone should be reasonably familiar at this point with the fine structure of DNA. We have phosphates alternating with sugars. In the case of RNA, these sugars are 5-carbon riboses. In the case of DNA, deoxyribose instead. We have nitrogen bases that are attached to these sugars that point inward. And remember that we have a sense of orientation with DNA, that you have a 5' end and a 3' end. Does everyone know that the 5' and 3' ends are named that based on the carbons in these sugars? Right, so you can see that this is carbon 1, 2, 3, 4, and 5. So the 5' end relates to that carbon number 5, and then the 3' end relates to this 3. So we see that we're always building in the direction of 3'. So the polarity that we have on the sequence and so on relates to that. I want to point out as well that some of the technologies that really dominated sequencing for forever were built around the idea that this attachment point is available for the addition of another nucleotide. When we use terminator sequencing, we are adding nucleotides to these sequences that block the further addition of additional nucleotides. So this is one of the ways that these that a lot of the sequencing methods we use create complementary sequences that don't cover the entire length, but stop at, say, the 30th position, or the 31st, or the 32nd. It's because they've incorporated a residue that blocks further additions. We have kind of a diagram of that here. You can imagine that you have this piece of DNA at the top that we want to sequence. So our process for doing so is going to involve building complementary sequences that cover different extensions of that. So you could imagine that the very shortest possible one you could incorporate would be just that very first nucleotide, the T. You might have another one that extends a little further, that includes just the last two T's, or the GTT, and so on. We're not directly controlling which strand gets a dideoxynucleotide, right? Our solution for performing this sequencing reaction includes both ordinary nucleotides and dideoxynucleotides. If by chance a dideoxynucleotide is incorporated, that prevents further extension of that complementary sequence. So we have a whole range of different products that are created in complement to the original template sequence. Now, I imagine at this point, you picked up this material back when you were undergrads taking biochem class, but it's important, I think, to remember that it's still very much live information. I would also point out that this is a rather different mechanism than what is typically used by machines like Illumina sequencers and so on. We frequently see a strategy where we're detecting the arrival of each nucleotide by its by a light that's emitted by the incorporation of a new nucleotide. So the upshot of this is this di-terminator sequencing gives us a whole series of complementary DNA pieces that contain varying numbers of nucleotides in that complementary sequence. Now, back in the old days, we would use a separation that would allow us to measure essentially the different complementary sequences that have been differentiated by just one nucleotide from each other. We'd be able to measure, basically interrogate which dye was incorporated in this complementary sequence and what is its size relative to all the other complementary sequences. Now, that might seem a little complex, so I want to try, we're going to talk about electropherograms in a little while that represent all of these different color traces that came back from the complementary sequences. Now, a lot of times, you'll find this called next-gen sequencing. But I would also point out that we've been calling it next-generation sequencing since I was a postdoc. And that's a very long time. At what point does next-gen get supplanted by the next-gen? It's kind of a bad name, right? So I'm going to refer to this as massively parallel sequencing instead. Massively parallel sequencing represents the case where instead of attempting to devote an entire capillary electrophoresis column or an entire gel lane to one particular template we're going to sequence, massively parallel sequencing is focused on sequencing millions of different templates all at the same time. So although the old school sequencers, like the ones you get from ABI back in the day, you might have had 384 capillaries. What that meant is you could sequence 384 different template sequences all at the same time. That's child's play compared to what you can do on your basic MySeq today. So we have lots of options for doing this, but all of these technologies really build upon the ability to sequence many templates all at once. Okay, so a lot of times these are working via the measurement of incorporation. That if you allow an A to attach to the growing strand, you're going to get a flash of color that represents A. If you allow C to be incorporated, you're going to get a flash corresponding to C, and so on. So this these approaches are not uniform. Like if I'm talking about a PacBio sequencer or an Illumina or whomever, or the thermo-ion torrent, they're all going to have slightly different technologies. But almost all of these give us the ability to create bazillions of copies of a particular template sequence, all located in the same very small location on this photocell that we're working within. So I may have the possibility of measuring up to 5 million different templates. I have 5 million different individual strands of DNA that have been sorted to a particular location in this space, and we've amplified to create a bazillion copies of it at that location, which gives us enough light that we can sequence that particular molecule. Okay, so we need to separate the templates, we need to fix them on a substrate. Typically we have some means by which we amplify, we create many, many copies of that template at that location, and then we're going to detect the incorporation of each base at each location. So I want you to imagine that each of these little blue plates is our photocell, right? Now, in reality, we're going to have millions of spots here, not just 4. But this little frame-by-frame cartoon is going to help us understand how we can sequence multiple things at the same time. So I want you to imagine that we're effectively working through a little cartoon. At the first time point, we incorporate nucleotides for each of the templates at each of the locations. And we get a little spot of green up here, a little spot of orange, or yellow over here, red over here, and blue over here. That tells us that the first nucleotide at this location is a T. The first nucleotide at this location is a G, and so on. When we incorporate this second nucleotide, we see that we get a flash of red up at the top rather than green, which is what we saw in the first place, or a blue at the left rather than a yellow at the left. The software remembers over time what color flashes have happened in what order. As a result, we can read this particular spot, the templates that are here at the left, as first incorporating a G, then incorporating a C, then incorporating a T, then a G, then an A. So the software infers that the sequence associated with this particular position in the photocell is GCTGA. So this is a little like watching a cartoon unfold. You know, you have like the first drawn cell, and your eye perceives motion because the second cell has the same objects just moved slightly, right? So in the same way, each cell of each incorporation step is going to produce a different color that's going to allow us to discern what sequence is present at that position. Okay. Now, I've already broken out a few of these terms, but I want to try to define some of them as well to make this a little more straightforward. So typically, we talk about the DNA that we want to sequence, the part that we're going to make a complementary sequence to, as a template. So that's a piece of DNA that we're going to sequence. Frequently, we are using something like paired end sequencing. We'll talk about that a little later in the slides. And this is a case where we have a long piece of DNA, maybe thousands of nucleotides, and the parts that we're going to sequence, we're not going to sequence the whole thing, we're just going to sequence the very ends, like the 5' end and the 3' end, we're going to sequence just those patches. So for that, we have a longer piece of DNA, often called an insert, from the days when people did a lot of like bacteriophage libraries and so on, often called an insert in that case. Now, we have as an input to the sequencer a template DNA piece, and the output is a read. That's the sequencer's interpretation of what it saw for that piece of DNA. We're going to talk about what these reads look like and the kind of information associated with them as we move through this first half. Frequently, you'll hear the term shotgun sequencing. Now, whole genome shotgun is quite a different strategy than the kind of sequencing we were doing back when I was in grad school. See, I was in grad school in the late 1990s, and we my lab was associated with the genome sequencing facility at the University of Washington. They had a very different notion about how to do sequencing back then, and the reason was sequencing was friggin' expensive. You couldn't just sequence everything you wanted to do. You could pick and choose what things you wanted to sequence, and you would get your 384 templates sequenced by one run of the instrument. And these were not cheap instruments, right? These were very expensive instruments. So the idea was sequencing is precious and expensive, so we're only going to sequence those pieces of DNA that help us to cover regions of the chromosome that we haven't already observed. That's a very different perspective than what we do now. So if you're doing whole genome shotgun, you have a lot less control over which pieces of DNA are going to get sequenced. But and generally speaking, you're dealing with sequencing reads that are much shorter than in the old days as well. So what you're hoping to do is effectively work with an old quote from, I believe, Stalin, who said, quantity has a quality all its own. That the old day sequencers, the old Sanger sequencers and so on, would give us very few sequences, but they were longer and they had better quality in general. Now, with whole genome shotgun, we assume that any deficiencies in our sequencing reads, they might be rather short because of the technology we're using, and they might have lower base pair quality. That because we're sequencing bazillions of them, that's going to be okay. We'll talk about how that plays out as we move into assembly. And finally, we have fold coverage. Fold coverage. You will find that if you go poking through the taxonomy at NCBI, the National Center for Biotechnology Information, that almost any animal you can name has had somebody try to do some sort of sequencing associated with it. We sequence everything. However, not all sequencing is done at equal quality. For example, if you were to look at an individual nucleotide in the sequence, the genome sequence reported for E. coli, you're probably going to have a higher confidence associated with it than somebody who has detected, say, cave bear DNA in a coprolite, right? They're sequencing at high quality and sequencing at low. One of the ways that we characterize the quality of sequence we have for a species is by fold coverage. So if you are working with a fungus that has kind of a very loose bit of knowledge about it, it was found in this particular sample, but we don't really have anything else in a similar genus and so on, and we've sequenced it at 2x coverage, it means if its chromosome is X, if its genome is X nucleotides long, we've produced only 2X nucleotides in sequencing reads. That's not very good, because if you do only 2X coverage for an organism, you are not going to have a complete end-to-end sequence for its chromosomes. Generally speaking, someone's going to have upwards of 10X coverage before they have what we'll soon call a scaffold that can cover a whole chromosome. So fold coverage is one of these terms of art that we use to describe how complete is the sequencing that's been done in this organism. So a lot of people will say, gosh, if we have massively parallel sequencers, if we have next-gen sequencers, why would we bother with the old school Sanger strategy? So let's talk about the old versus the new. Generally speaking, when we talk about capillary electrophoresis, fluorescent tag, automated sequencers, the kinds of things that completed the Human Genome Project back in the late 1990s and early 2000s, we would see that an individual template would be sequenced to a length of about 600 base pairs. 600 base pairs. Today, it's very frequently the case that our next-gen sequencers are cranking out about 150 base pairs instead. Now, some of them can go much, much longer than that. There's a lot of reason to think that when you're sequencing a novel organism, that doing so with a combination of very long sequences and a massive number of very short sequences is actually a nice gain, because the very long sequences help you with scaffolding, whereas the very short sequences, by the very large numbers, ensure that you have the nucleotide sequences correct. So in general, when we talk about stuff like Illumina sequencers, we're talking about relatively short reads. Generally speaking, we would have a greater sense of accuracy about individual reads in the old school Sanger sequencers. But, as I said, there's a quality to having very deep coverage that results from having lots and lots of templates sequenced. So by playing those off of each other, you're able to compensate for the relatively high error rate associated with these shorter reads. Because we're able to sequence from millions of reads rather than tens or maybe hundreds, maybe a couple hundreds templates, we find that massively parallel sequencing gives us oodles more information to work from. It also means, however, that we have a considerably more difficult time storing all the data that come back from the sequencer, and quite a lot more bioinformatic challenge in making sense of them too. So there's a lot of weight placed on bioinformatics by the fact that we have these short sequencing, massively parallel sequencers in play now. Now, how many people have seen an electropherogram at this point? A few people have, okay. Often an electropherogram is going to be apparent because you've done some sort of massively parallel sequencing experiment, but you've added on a confirmation of a few questionable sequences through Sanger. So you might find that you need to be able to interpret both kinds of data. Electropherograms in general are not something that we retain when we use a short read Illumina sequencer. They're just too much, it's way too much bulk for us to do so. So frequently what we'll store is not this electropherogram, but rather the information we derive from it. You have to pick your battles sometimes, right? If you find that storing a 1 gigabyte file for a sequencer is painful, try storing 10 gigabytes per file. It doesn't get better. All right, so this is an electropherogram, as I mentioned, and each color that you see here, black, blue, green, and red, represents a different nucleotide. So if you see at a particular location that you have a tall black peak, but there's no green peak, no red peak, no blue peak behind it, you can say, this is unambiguously a G. If you have a nice tall red, you can say, that's a T. If I have a nice tall blue, it's a C, and so on. It might seem that reading this is really, really simple. If I just sit here with I don't have a pointer stick, do I? That's okay. No, I've got this guy right here. He's now my pointer stick. Let us imagine then that we have red, black, green, 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 blue, black, green, red, etc. We can walk our way across there, no problem. And how confident are you going to feel about, let's say, this little nucleotide right here at 450? Okay, I'm claiming that this is a green peak. Therefore, I'm inferring that there's an A there. What's my chance of error associated with that? Are we in coin flip territory? Am I more confident or less confident than a coin flip? No one's really sure on this one. Okay, I'm going to claim that this position at 450 looks pretty good to me. I've got a nice strong green signal. There's very little other color that's getting a bump at that spot. I feel pretty sure that this is 450, but I need to quantify that. I need to pin it down. I'm going to argue we're much better than coin flip territory. I'm even going to say we're better than 1 in 10 chance of better than 9 in 10 of getting it right, or less than 1 in 10 of getting it wrong. But am I capable of pinning down whether that's a 1% chance of error, or a 1 in 1,000 chance of error, or a 1 in 10,000 chance of error? That's difficult, isn't it? So when we read an electropherogram, our goal is not just to come up with a sequence of letters. It's to come up with a probability of error at each of these locations. This is where the Phred algorithm comes in. We're going to spend some time talking about how that works. I want to point out some other things. I pointed at 450 because this region looks pretty good, right? But what happens when I slide to, say, this region at 720? Do you see nice and sharply defined peaks? How many A's does this long stretch, this long plateau of green constitute? Is that 4 A's? Is that 5 A's? Is it 3? We no longer have good resolution, do we, to say this peak is separate from this peak and so on. So this is something that's pretty much common to all chromatographic methods, that anytime you're doing a separation, your ability to separate these species from each other and unambiguously depends on how big they are compared to how big the difference in mass is, right? Or whatever property you're using for separation. As you're separating these nucleotides through capillary electrophoresis, your ability to tell something that's 700 nucleotides in length, apart from something with 701 nucleotides, is pretty small. Okay, likewise, look at the stuff at the beginning of the sequence. Would you feel comfortable trusting any of the base calls before 50? No, it's a big smear, it's a mess. So we have kind of a sweet spot in the middle of the electropherogram. We have problems of resolution at high base counts, and a whole bunch of smear down at the bottom. That's not a very good place to work. So being able to say which parts of this read are viable is also something we need to be able to do. So yes, we need to be able to infer a sequence from the electropherogram. That's the base calling algorithm. Phred is going to associate with each base call, with each letter, our probability of error for that letter. So how do we discern a good base call from a bad? There are several things that come into play here. So first off, we want that peak to match the beat of its neighbor. We want to see only one trace at that location, right? If let's say this is the black peak, we want there to be a black peak and no red peak and no blue peak and no green peak. We want just one color to go up and down at that location. And we want the highly probable errors to be far away. So I'm going to tell the story briefly. I've given myself a lot of slides, so I can't tell you many stories. But when I moved to Nashville in 2005, I moved to an area called 12 South. Now, at the time I moved there, 12 South was an up-and-coming neighborhood. The realtors are like very excited about up-and-coming neighborhoods. It has this, it's a neighborhood that has artists living in it, and it's got up-and-coming, and you're close to town, and so on. But it is also, what in the States gets lambasted as gentrification, which is to say that a bunch of folks could no longer pay property taxes because their house values were suddenly soaring compared to where they were living, which were often pretty lousy houses. So I moved into this place, and it was gorgeous. Gorgeous! The kitchen had been redone, nice marble top, lovely island in the middle. The floors were new and wood and gorgeous, and it was so nice. So nice. And then one day, I was wandering around in my yard, and I noticed a syringe down near the foundations of my house. And I asked my neighbors about it a little bit, and they said, "Oh yes, your house was the crack house." The house I moved into before I lived there had been the place where all the drugs were dealt in my part of the neighborhood, in the 12 South neighborhood. Some flippers had bought the house at low price, torn out the insides altogether, just left the frame there, and then rebuilt a house on that frame. But if I'd seen my house even 2 years before I bought it, I would not have felt comfortable living in that neighborhood at all. Right, so let's imagine the 12 South neighborhood then, as it was before I moved there. Imagine Dave's house is the epicenter of drug sales for 12 South, right? Now, if your house is next door to the crack house, is your property value high or low? Now, was my neighbor selling crack? My neighbor was not selling crack. They're very nice people. But they were suffering because their house was next door to the crack house. What if you were a block away from the crack house? Is your property value diminished or okay? It's still pretty bad. If you live a mile from the crack house in the very tony uphills just south of Vanderbilt University, is your property value diminished by being within a mile of a crack house? It's not. In the same way, if you're in an electropherogram and you happen to be very close to a bunch of stuff that's unresolved, that's got lousy quality, your base calling error probability is high, even if this peak looks like a very pretty peak. Proximity to something bad can tar you with the same brush, to use the phrase. Okay. Now, I've also made a lot of mention of a beat, of a particular frequency associated with those calls. I'm going to just, I want you to imagine this not as an electropherogram, but as the signal on the equalizer in your stereo when you've got the sound turned way up, right? So you're listening to something that's got a very distinctive beat. Do people listen to house music anymore, or is that a completely over thing? Maybe dubstep. How about dubstep? All right, what is the characteristic of music of the house genre or of dubstep? Just say it.
Very rhythmic.
A very rhythmic beat, okay. And by very rhythmic beat, we mean a bump, 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 bump. Okay, that is a frequency that we've got going. So I want you to imagine now that this is time, and this is the amount of sound coming out of the woofer on your stereo, right? So, womp, 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 womp. Just as is true in your dubstep music, you want these beats of the electropherogram to be spaced equally far apart. That we've got the black step, the black peak here, the blue peak here, the red peak, the green. We expect these to be spaced equally as a function of time in the capillary electrophoresis. We associate it as with a good thing if this beat is preserved, and we feel bad about it if it's not preserved. Almost any time in bioinformatics that the phrase frequency or beat shows up, it's not long before someone's going to mention Fourier Transform. Does everybody remember Fourier Transform class from any undergraduate engineering classes? Sadly, many people in our field have never taken an engineering class. And that's just sad because they have an awful lot of useful tricks for us. A Fourier Transform is a way to transform an amplitude as a function of time, in this case the the fluorescent intensity as a function of capillary electrophoresis separation, to translate from this amplitude over time into a frequency signal. So if we look at this signal, we see that it's got a pretty good magnitude, about twice that of this lower signal, and it's spaced pretty far apart. So in the frequency domain, we see that we get a peak here and here to represent both the positive and negative versions of that frequency. This one has a higher frequency. You see why it crosses the zero line more times per unit, but it's at a lower magnitude. So it's going to be a smaller peak in the frequency domain, and it's going to be at a higher value. So if we saw a signal like this that represents the summation of this signal and this signal, we could decompose it to be the combination of those two frequencies. In the same way, and I have to say, Fourier Transform is used bloody everywhere. If you have been listening to your iPod this morning or whatever, you've been making use of Fourier Transforms. So stuff like MP3 encoding makes use of Fourier Transform as a really powerful way to compress music into files. In the very old days, before we were using stuff like Fourier Transform in our encoders, compressing music was a really dismal affair, I have to say. Things have gotten a lot better since MP3. So, if we are looking at the signals that we see in an electropherogram, we can decompose those to a base frequency at which we expect new nucleotides to appear by using Fourier Transform. Okay. Now, let us imagine that the Fourier Transform has given us an expected rate at which new nucleotides are appearing. That appears along this numbered timeline at the top. Predicted location, predicted location, predicted location, predicted location. The period, the time between beats on our electropherogram is what's fallen out of the FT of the electropherogram as our set as the key rhythm at which new nucleotides are coming. So we now have predicted locations where peaks should be, and we have the observed intensity that we've observed in each channel. So when we look at a particular peak, we might find that there's a bit of shift between where we expected that peak to appear in time and where it really did. And in this case, we want to punish those nucleotides. We're going to say those nucleotides are a little misshapen. They're a little earlier or a little later than we expected them to be. As a result, we have a higher error probability associated with it. Okay. So, we now need a way to express numbers that are very, very small. As you saw from that electropherogram, most of the peaks are pretty beautiful, really. I mean, most of them are right where they expect them to be. There's only that one color and not another color at that location. Its shape is pretty nice and concave down. It looks like a nice peak, etc. So we need to be able to talk about vanishingly small differences in the error rates for these peaks. And this strategy was used in Phred, but it gets used in all kinds of different places. This negative log expression of small numbers is very, very widely used in a huge array of bioinformatics tools. So whether or not you need to ever implement your own Phred, understanding how Phred expresses those probabilities is very important to you, to understanding almost any bioinformatics algorithm. So we now have a probability of a base call error at this particular letter of sequence, and it ranges from 0 to 1. 1 meaning we're absolutely sure this is wrong, and 0 meaning that there's no chance that this is wrong. So the proximity to 0 matters quite a lot. Being 1 out of 100 is really different than being 1 out of 10,000. Could we agree that? All right. So I would point out that the Human Genome Project was funded to produce a genome sequence that was correct to less than 1 in 10,000 chance of error at each location. Right? So if the senators are only going to give you money if you produce a product that's been quantified in this way, you better have a mathematical way to talk about that. All right, so what we use then in Phred is a Q value here that represents -10 multiplied by the log base 10 of a probability. That does not inherently make a lot of sense for people, so I've given some examples here. So let's start with a probability of 0.01. This is an error probability of 0.01. Does somebody want to put that into words for us, what it means? It's not a p-value, it's a point probability. The probability of error at this base is 0.01. What's it mean? That's very good, yeah. It means that there's a 1 in 100 chance that the software has miscalled this base. Great. So probability of 0.01, if expressed in scientific notation. Everyone remember scientific notation? That's good, that's good, all right. 1 times 10 to the minus second power is 0.01. If you were talking in percentage, you would say it's 1%, a 1% chance of error. In Q, that gets expressed as 20. So 1 times 10 to the negative second, taken in log 10, is -2. -2 times -10 is 20. The math there might feel a little fuzzy, but it's going to get a lot clearer, I think, as we walk through 2 more probabilities. A 1% chance of error, a 1 in 1,000 chance of error, a 1 in 10,000 chance of error. So as we do that, we see that we go from a Q value of 20 to a Q value of 30 to a Q value of 40. So at each stage, as we get 10 times less likely to have an error, we see that the Q score, the probability, the Phred score associated with that base rises by 10 in each case. Okay, so if I report a base from my sequencer with a Phred score of 10, how do you feel about it? What's the chance of error for a Phred score of 10? 1 in... Okay, we'll work our way backward. We have a Phred score of 20 at a particular nucleotide. Do we trust the nucleotide? It's got a chance of error that's 1 in...
100.
100. Okay, we said that 20 corresponds to an error of 1 in 100. So a Phred score of 10 corresponds to an error of 1 in
10.
1 in 10. Okay, that sucks. We do not want to deal with anything that's got a Phred score of 10. All right. Generally speaking, you will find that a lot of people end up using thresholds of 30 for Phred scores in sequencing experiments. That they just trim out anything that falls below with a lower Phred score than that. Which means that they're throwing out any base calls that have a chance of error greater than 1 in 1,000. So that's pretty restricted, actually. But because we have so many sequences to work with from a modern sequencer, we can do that. All right, so we see that the number for Phred, the Phred score gets higher and higher and higher. It rises by 10 for every 10 times less likely error is. A high Phred score means a very low probability of error. And yeah, if you get below 20, don't even think about looking at that nucleotide twice. All right, when we run a sequencing experiment over at the facility, you're going to get an output to work with. And typically, that is an output in FASTQ format. FASTQ is a very widely used format, and I want to note another weird computer sciencey thing that's going on here. So if you look at the top line in this little segment of 4 lines, the FASTQ file basically digests into 4-line chunks. So this first line, starting with an @ symbol, we're not making a direct call out to somebody on Twitter. The @ symbol here means this is the address of this particular sequencing read. All right, so it's got a nice long sequence associated with it, but then here we see that in line 2, we have CCC, GCA, CTT, blah, 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 all the way down to GTT. Great. So that is the sequencing read. A FASTQ file is just a really big text file that's comprised of block after block of these 4-line segments. Each read is going to correspond to a 4-line block. So the sequence appears in number 2. The first one's just the address. The third line, typically, is just a plus symbol. This comes from the fact that we used to have a duplication of the address from the first line to tell us where this line of quality scores comes from. But in practice, it's just a plus these days. We then see a whole bunch of other letters. Now, A, what is all this business? Are those nucleotide base calls? They are not. Has anyone heard of ASCII? People have heard of ASCII. Oh, this is good. Okay, ASCII is the predominant way that we tell computers what number for a byte corresponds to a particular letter. So the letter capital A, for example, is 65 in ASCII. It's just by convention, really. The capital letter B is 66. The capital letter C is 67, and so on. All right, so these letters, in fact, represent numbers. We can store up to 255 down to 0 for an individual byte. So the A's are 65, the E's are what? Let's see, that'd be 4 more, so those would be 69. But we're not using these numbers as they are. The way that we report quality scores in FASTQs has a certain correspondence between the letter and the Phred score. Because the first letter we use is not 0 in ASCII, it's bang. So in effect, the bang represents a score of 1. And by the time you get to capital A, you're at a score of 32. So in this case, the software is saying that the first base call is C, the quality associated with it is 32. The next C corresponds to a 32, C, 32, G, 32, C, 32. This next A, however, gets a score 4 higher than that, 36. Are those acceptable base calls? For all of them, the chance of error is less than 1 in 1,000. So yeah, this is a pretty good read, in other words. When we grab these from the sequencer, the text files are absolutely massive. And you will frequently find that people have zipped them up in some way. So you might see an extension of fastq.gz. This is a zip that was created by gzip. Or you might find something like fastq.bz2, something created by blockzip. So we have lots of options for making these really large files squeeze down much better. Frequently, to shove it through a bioinformatics tool, though, you must unzip the file, which means you better have a lot of disk space ready. Okay. Graphically, one of the most common ways that we perceive this is through the FastQC software. This is created by Babraham AC UK. You can download the new version. It's free. And so when you are given a FASTQ file, if you want to evaluate whether this was a successful or a failed sequencing experiment, FastQC is your way to get there. So in this case, we are looking at quality, a box plot of quality, as a function of where in the read it is. Now remember, with old school sequencers, with Sanger's, we might have as many as 600 base calls within a read. In this case, we're using next-gen sequencers. We're using massively parallel sequencers that are generating only 40 nucleotides for each read. And you can see that at the start of the run, our median base call chance of error is above 39. So that's really bloody nice. By the time you get to the end of those 40 nucleotides, though, you've accumulated a greater and greater amount of error. And so our base call probability, our error probabilities have risen a bit. As a result, Phred score has fallen. So I'd love you to just have that relationship in mind. I mean, we talked about the math of it, but generally speaking, a 30 is a pretty typical kind of error quality, error probability associated with a base call. As errors become more likely, Phred scores fall. As errors are less likely, Phred scores rise. So that inverted relationship is something you definitely need to have in mind. FASTQ is just a way of visualizing across bazillions of reads what the error probabilities look like. All right, now we will have 5 minutes in which I rest my vocal cords slightly, and we can take quick bio breaks, but then we're right back. Welcome back to the second half. We are going to talk about sequencing informatics. Having already had some time to discuss the technologies for high throughput sequencing, we've talked a lot about Phred scores. Obviously, that's one of the big takeaways that I hope everyone is able to work with in this course. For the second half, we're going to move on to how we make use of this information in the case of mapping and assembly. So we're going to start by these two chief uses and try to dissect them at a very high level. First off, mapping is an approach that's relatively information dependent. If you are doing sequencing for the first time in a non-model organism where we don't have a genome annotation to compare to, mapping is not really an option for you. So if you have a known genome annotation, mapping is a really good option for you. Typically you'll find that mapping comes into play when we do stuff like sequencing messenger RNA from humans, RNA-seq for example. But there are lots of cases where you would be doing this mapping as an approach to look for sequence variants. For example, the people at my university do a lot of sequencing of Mycobacterium tuberculosis. We've known the genome of that organism for a long time, but we do sequencing of it because we want to learn about which sequence variants are present in a strain of tuberculosis that's circulating in the population. Because some types of Mycobacterium tuberculosis are very good at resisting antibiotics. And by sequencing these organisms, we may find sequence variants that cause a given strain to be more proof against this kind of cure. So in a lot of cases, this mapping is a good approach. So just as a point of perspective, you might expect that a given sequencing experiment for a human could be mapped in about 15 minutes, say. That would be a pretty good outcome against the human genome annotation. Very frequently, we are making use of something called exome sequencing, and that's an obvious case where we would want to do mapping rather than assembly. In an exome, we are sequencing not random patches throughout the genome, but just patches within the expressed genome, which is to say the spots that code for messenger RNA typically. So this mapping approach is a really dominant one when you have a very well annotated genome. Frequently, however, we are working with species that have not been adequately annotated at the genome level. For something like this, we're quite likely to make use of something called assembly. Here we are trying to build contigs. Is another one of those key terms. We're trying to build contigs that represent places where these reads overlap with each other. And hopefully we can make large enough contigs by stitching together overlapping sequences that we can get a very good large-scale sequence of the organism derived from the overlaps among reads. If you're working in non-model organisms, this is pretty much your only bet. That we don't have a good enough annotation for the genome for the species, therefore we must rely upon de novo assembly, as we frequently call it. De novo meaning new, that we're creating a new assembly from new information, basically. All right, so why do we emphasize mapping so strongly? First off, doing assembly takes a lot of time, and it takes some pretty expensive computer hardware. Now, computer memory gets larger all the time. I mean, I think it's quite rare these days that you'd buy a good laptop and have less than 4 gigs of RAM, for example. That's an unthinkably large amount of memory for someone who was doing a computer 10 years ago. However, when we typically make use of high-end workstations for doing assembly, we need to have, maybe 256 gigabytes of RAM to do a proper de novo assembly. That's a pretty heavy bit of hardware. You can do it today with even commodity hardware, but it's relatively rare that you run across computers with that much RAM for other purposes. Doing a mapping, on the other hand, is something that you can do rather efficiently and quite a lot less RAM. Being able to recognize sequence variants is a very, very popular use of mapping technology. So when you have an annotation, you probably have some sense of what the wild type sequence is for the species. And from that, you can recognize which sequence variants are present in a given sample. Today, massively parallel sequencers like Illumina's produce more than 100 gigabase pairs of sequence a day. It's an astonishing volume of data. So if you're going to be producing data like that, being able to use a relatively lightweight bioinformatics workflow to make sense of it makes all kinds of sense. If you're trying to do a de novo assembly from each data set produced on an instrument, you're going to need an awful lot of computers with an awful lot of great specs. So generally speaking, when we get downstream to the tools that, for example, produce variant call files and things like that, doing things like FASTQ, trimming out parts of the reads that aren't all that useful in terms of base quality, and aligning to a reference, i.e. mapping, are expected for any downstream tools. So it's generally the case that if you can map, you will map your data before you do any of the advanced applications from that. One of the biggest approaches used in this space is the Burrows-Wheeler Alignment Algorithm, or BWA, you'll frequently see it called. Now, the two people whose names I've called out here, Burrows and Wheeler, are actually really, really famous, but they are not famous for the Burrows-Wheeler Alignment Algorithm. David Burrows, as I recall, was the inventor of this little thing called a subroutine. He was one of the creators of this foundational programming strategy that we use for separating big chunks of code into those that can be decomposed into smaller subsections of code. So, very famous computer scientist for obvious reasons. Wheeler, on the other hand, was very famous about the time that I went to grad school. Before everyone used Google for everything, you frequently found people using things like Digital's Alta Vista search engine. Well, he's the guy who wrote that. So these guys are both really famous in the space of computer science, but their ideas turned out to be very directly relevant in the space of next-gen sequencing. In particular, the suffix array gives us the ability to do very fast mapping. So I want you to think for just a moment about how you would go about mapping an individual read. Right, so you have one read, maybe it's 50 nucleotides long, and you have a human genome annotation. The human genome is 3 billion base pairs. So imagine that you have a Word document that's 3 billion base pairs. How would you go about mapping an individual sequence read to that? I already started us with Microsoft Word as our engine, right? So find, I think I heard someone say, right? There it was, fine, perfect. So you'd hit Control F, type in the read sequence, and hope that somewhere in the massive genome annotation of 3 billion base pairs, you were able to find this 50 nucleotide read. That's a mapping experiment. But you see, it's going to take Microsoft Word a little bit of time in order to sniff through a document that large for every place where this particular 50 nucleotide sequencing read maps. Right? That's going to take some time. And I note that our sequencer has kicked out millions of reads, each of which needs to be looked up. So having to look inefficiently in this way, having to sniff for read number 1, read number 2, read number 3 is kind of pointless. A lot of these reads are going to cover the same regions. So the suffix array gives us the ability to make one structure that we can compare the entire database to in one pass. The suffix array allows us to turn each of these reads into suffixes. So all of these different subsequences that start at the 3' end and go back some number of nucleotides. Once we have done this for one read, we add the next read to our suffix array, and the next read. We keep adding to the suffix array all of these different suffixes derived from our read links. The other thing that we're going to do is sort the suffix array by the sequences inside it. This might seem like a very bizarre thing to do, but consider the fact that we have many reads that overlap within the body within the genome of the species. So here we have this particular long sequence in the suffix array. There are about a bazillion reads that contain this particular thing. We don't actually care at this point how many there are. We just want to know where the reads we have align to this reference sequence. So Burrows-Wheeler is an amazing strategy because through bisection, we're able to say very quickly, we spoke about bisection a couple days ago when we were talking about looking through a sorted list for a particular number. You can use the same trick. You can do bisection of all of these reads that you've generated to determine whether an area of the genome matches to that. This is one of these cases where searching and sorting really comes to the fore in making a very complex task tractable. By putting all of the reads together, we make it possible to compare in one pass to the genome to say where any one read can match. Kind of astonishing. So that is one of the canonical tools that we use for this read mapping approach. Now, generally, what comes out of something like this is a BAM or a SAM. We've already talked about FASTQs. FASTQs are what came to us from the sequencer, right? The FASTQ provided what kind of information to us? Base calls. It told us what letter is at what position. And it provided us Phred scores, quality scores to say what is the probability of error at each of these reads. Now a BAM is quite different because it reflects the result of a mapping. So having a BAM file means being able to say what reads did I detect that overlapped any exon of this protein coding gene. That's pretty powerful, isn't it? Right? So this represents a great consolidation of the information that we've produced through mapping. The text that we find within SAM and BAM files, well, a SAM file is the text version, a BAM file is just the same file in kind of a binary format, is that we have records that have a read name. Remember, you have to have some address that you use to describe each read. It has a position to say where within this genome was this aligned. It has stuff like map quality to be able to say, now this is like Phred, but it isn't the same thing as a Phred score. Remember that Phred is saying, what is the probability of error at this position of at this base call of the read? But a map quality is different. It's saying, how much can I trust that this whole read links to this position in the genome? So it's a mapping quality reflects many bases, not just one base. That's a difference. It has the sequence that was produced on the read that we've now aligned to this. It has quality scores relating to that, and it has some additional metadata as well. So we have a great deal of information that's then expressed about these reads, and it makes it possible to say where in the FASTQ files can I find the reads that relate to this gene. But in this case, you can ask that question of any gene in the genome. Very powerful. SAM and BAM. All right. Now, sequence assembly is kind of a different class of problem. We've got bazillions of short reads. Bazillions is a technical term. And what we want to produce are something that we call scaffolds of contigs. Scaffolds of contigs. Ideally, when you have something like a finished annotation, each chromosome represents one scaffold, where you can say that all these different patches of sequence that I know, spanning from one end of the chromosome to the other, you're almost always going to have something you're missing. So, we have highly repetitive DNA regions. We've got centromeres and telomeres. These are areas that are really tough to sequence. So it's quite unlikely that you're going to have one contiguous sequence from one end to the other in anything but a model organism. So in this case, we're trying to figure out all of the different contigs, regions of uninterrupted sequence, and how they relate to each other, the scaffold that relate to each of these chromosomes. You might think that mapping would take care of everything, right? If you're producing a new sequence for a human being, you might think to yourself, well, why do I need de novo assembly? This is never going to come into play. The reality is that when you do a mapping, even in a human being, you're frequently going to find that some fraction of your reads cannot be mapped. They're not exact matches for what shows up in the genome. And you can allow for error-tolerant mapping to say, well, this could be a C or a G. Everything else of this read maps perfectly, so this is clearly where that G belongs. But generally speaking, there's always going to be some fraction of the reads that aren't going to map for you. And for that, doing a de novo assembly is often very helpful. What if you've sequenced a human sample, but you also had viral proteins in there? Viral DNA in there. A situation like that is going to be problematic because a DNA read that corresponds to the viral DNA is, of course, not going to map to the human DNA. So you might do de novo assembly to see, gosh, what else is in here that is causing all of these unmapped reads? Okay, so of course, there are lots of people who work in organisms that don't have mature genome annotations, and for them, this kind of de novo assembly is just required. All right, so I've already mentioned the terms contig and scaffold. Here I'm going to try to define them a little better. I'm using some definitions that I borrowed from this webpage here. A contig is a contiguous length of genomic sequence in which the order of bases is known to a high confidence level. So I think of this as a contiguous region where every single nucleotide is known with great confidence. But that is, it is plausible that in some cases people would use a weaker definition than that, where gaps, for example, are allowed to say, well, there are 3 nucleotides here, but I'm not sure what they are. Or there's some number of nucleotides here, and I'm not sure what they are. Scaffolds, generally speaking, are composed of these contigs that we've produced, along with gaps. And one of the valuable things that a scaffold is able to tell us is the orientation of these contigs with respect to each other. So you can think of a scaffold as a way to stitch together a bunch of contigs, and it may reveal to us regions of the genome where we need more sequencing data to do what's called gap filling, to fill in that space. Okay, so the ordering of contigs should be known. It's contig 1, then it's contig 2, then it's contig 3. And their orientation with respect to each other will also be known when we have a mature scaffold. I see a question for me. No question for me? Okay. All right, so let's imagine a scenario where every read is independent of every other read. This is not the ideal scenario. This is just kind of a what-if question. We started with a biological reality, the genome. In this case, maybe we're looking at an individual chromosome, shown in gray up at the top. We have reads that have been produced at random positions within this chromosome, and each of those has now been sequenced. You can see that the reads in that first pile of 5 all overlap with each other. So we are able to project downward through these reads to create a contig that starts at the leftmost location, the 5'most location, and extends up through the 3'most location. This contig reflects the overlapped sequences stretching from here to here, in other words. This second contig is working from this set of 5 that have been overlapped. Now I note that this read and this read do not overlap each other. We see that the last position on this one doesn't touch the 5' position on this one. And yet, because these two both overlap that sequence, we know that they do belong next to each other. Okay, so we can project downward, and from this we get a contig, and from this we get another contig. If we have some other information, something like, in the old days, like a restriction fragment length polymorphism map or something, we might have external information that tells us this sequence is next to this sequence in the genome. But we have this problem that if we're trying to create a scaffold that positions these two contigs together, we really need to have some kind of information in order to justify that they come close to each other. This is difficult when we're dealing with short reads, a short read based sequencer, which is to say that the patch's individual reads cover is short, and if we're doing a case where every read is independent from every other one. When I was in grad school, I met a guy named Jared Roach, who was a PhD student, a little older than me. And I always thought of him as this kind of funky guy who wears bow ties. But in fact, he created he produced one of the seminal papers that describes how we do sequencing, whole genome shotgun sequencing today, the paired end read strategy. So I want to try to explain what a paired end is before we move much further on. Imagine that I have some insert sequence. Now, they're called inserts in this case because of the kinds of libraries from which they were built and so on. But imagine that I have a long piece of DNA, not terribly long though, maybe 10,000 nucleotides, right? That's not big. So 10,000 nucleotides. At the time, we're always talking about sequencers that could produce at the most, say, 600 nucleotides of sequence. But we wanted to avoid the problems that we talked about in the prior slide, that we wanted to be able to know which which reads happen to be close to each other. So in this case, if we have an insert that we've grown up in some vector, we know what vector sequence appears on this end, and we know what vector of sequence appears on the other end. We're able to create these primers then that are used to create where these read sequences will come from. So this insert of sequence might be drawn from a random location in the genome, but we know that a patch of sequence that's read from its 5' end corresponds to a piece of a read that's generated at the other end of that piece of DNA. So one piece of DNA, two patches of sequence, but we know that these two are located next to each other within a few thousand nucleotides, and that they point in opposite directions, that they're on complementary strands. So I would remind you that we're building contigs out of overlapping sequences. When we draw in this diagram in kind of light blue and dark blue patches, the dark blue patches are the parts that we're actually sequencing. They're the reads. And we draw in a light blue line to reflect that they come from the same piece of DNA. They are paired end sequences. I was really happy that Jared was willing to provide me the original image he used in his paper, so here we have it again. So we see that some of our inserts are relatively long. That's not to say the reads are long. That's to say that the piece of DNA from which the reads were produced is long. So we can still project downwards and say, if these reads overlap to a high degree, then we can stitch them together to make a contig. But we have something really powerful here in that we're able to say, if this contig includes this end of the insert sequence, and this contig incorporates this end of that same insert, we know that they're close together. And we know how they're oriented with respect to each other. So now, although we may not know even exactly how many nucleotides fall between them, we know that they're close together, within a few thousand nucleotides. And that is a lot of information to work from when you're trying to build a scaffold from these contigs in the end. So is there a question before I move ahead? Yes?
Sorry, but what happens in the case if there are long stretches of identical sequences in one target sequence?
We're going to talk about the problems of junk DNA in just a minute, which relates specifically to that question. So we'll come to that very shortly. In fact, it's the next slide. Good question. So we have lots and lots of DNA in our genomes that is a bit of a relic of a bit of a war between our DNA and some other DNA. So these transposable elements are pretty good examples of this. Has anyone heard of ALU sequences? ALU sequences. They're called ALU sequences because there's a restriction endonuclease that cuts at this sequence, and we discovered that ALU cuts lots of places in the human genome. ALU is a primate-specific, short interspersed repeat element. And ALU shows up, is still able to make new copies of itself in our genomes, although it's fairly rare. Every 20 births, there's another insertion of ALU. But our genomes have gotten pretty good now at defending themselves from the insertion of more ALU elements. That's just one of several different kinds of elements that we have. As a result, we have many, many, many copies of these pseudogenes and transposable elements and so on that appear throughout our genomes. These cause a real havoc when we are trying to assemble whole genome sequence. So I'm going to just plow right ahead. I want you to imagine a scenario where the blue gene is close to the green gene, which is close to the, shall we call that cornflower? The cornflower gene, which is quite close to the yellow gene. I'm not sure the yellow one is all that easy to see. So we have 4 genes, and they're all close to each other. In between them, we have a particular repetitive DNA sequence that looks like, that sits in between all 4 of those genes. Everyone has that so far? When we produce sequencing reads, the reads are so short that they don't generally extend from one color gene into the next color gene. They're much shorter than that. So when we produce a read, it might be only from the dark blue gene, or it might be only from the green, or only from the light blue. But often we're finding in this case that they extend all the way into the adjacent repeat sequence. The problem here is that I might think the dark blue gene is next to the cornflower gene because they're both next to a repeat that has a really similar sequence between the 2 locations. That would be a mistake. They are not. But we might make the mistake of thinking so. When we make a layout graph, each of these reads gets represented as a dot along this path. And we're going to draw edges to say, where are there overlaps that would be permissible moves from this read to the next read? Do you see that? When we have something that's all blue, it can be next to, it can be adjacent to, something that has elements, that has the 3' end of the dark blue gene and the 5' end of the repeat sequence. So we could move from here to there because they overlap in sequence. When we get to DNA a DNA read that's produced only within the repetitive DNA, however, we have this real problem because there are lots of transitions we can make from there to step from the repeat to either the green gene or the cornflower gene or the yellow gene. That's awkward because now we have this problem that many different transitions are possible leading out of this, and many of those paths are wrong, only one of them is going to be right. So these edges represent overlaps between reads that we could follow in order to stitch together a larger sequence. But repetitive DNA is a huge burden on this. From this, we eventually want to create a de Bruijn graph. We want to be able to say this blue gene leads into repetitive DNA, which could then lead into the green or could then lead into the cornflower, but which finally lands us in the yellow area, after which there is no repeat. These graphs are our way of telling us about permissible paths to transition among the reads to get through. This approach used to be built around the idea of Hamiltonian paths. I'm not going to ask you to define what that is, but I think it's good for you to have heard the term. A Hamiltonian path says, we generated all of these reads from the same DNA. Therefore, we need to find the path that uses all of the read information we produced exactly once to produce one big assembly. This, it turns out, was a very problematic way to go about making de novo assembler algorithms. Now, a lot of the new tools have run into the same problem of repetitive DNA. So here's another way to visualize the repeat problem from Arachne. This Batsoglo fellow, for his PhD project, wrote a whole genome assembler and demonstrated its use on a whole genome shotgun set for Drosophila and assembled it on a single student's PC of the time. It's an astonishing piece of work to have created something this complex in a PhD thesis. So imagine that you have one region of the genome in which the gene A is next to a repeat, and then that repeat is next to B. From that, we have a read, here labeled X, that starts in gene A and continues into the repeat. In another area of the genome, far away, we have two genes C and D. They're separated by a repeat. Now we have a read Y that runs from the repeat into gene D. If all you're looking at is the read X and the read Y, you might think, I can overlap these two, and now you've made a critical mistake, because your assembly, the contig you're generating, makes it look like gene A is close to gene D, and they're not. So repetitive DNA is a really big hassle in situations like this. So K-mer catalogs are one of the ways that we try to efficiently deal with the overlaps. Does everyone know the handshake problem? Okay. If you have three people at a cocktail party, it's a very small party, a cocktail gathering at that point, how many different handshakes can you have between person A, B, and C? Lots of different answers there. Okay, we'll walk through it. Can person A shake person A's hand? Okay, you can't shake your own hand. We're going to rule that out. All right. Person A can shake person B's hand. Does person B then also need to shake person A's hand? Okay, so we have commutativity, that person A having shaken B's hand is equivalent to person B having shaken person A's hand. All right, so A can shake B, A can shake C, B can shake C. Just 3 handshakes possible when you have 3 people present. But it gets harder to walk through all those possibilities if you have 5 or 10 people attending the party, right? This is called the handshake problem. And the way that we express the answer is that each person...
How did you even know that happened, by the way?
What's that?
How did you detect that like that quick?
Oh, it made a little ping ping sound. It's okay. All right, so the way that we resolve the handshake problem is that each person can shake n-1 handshakes at a party, right? So if you have n people, each person can shake n-1 handshakes. But we also have to deal with this commutativity problem that if A shakes B, B shakes A. So the way that the math works out is that n, the number of people at the party, times n-1, the number of handshakes possible for each person, divided by 2 gives us that number. So if you have 10 people at a party, the number of handshakes you can have is 9 times 5, 45 different handshakes, right? I want you to imagine the problem, though, when we're dealing with sequencing data. If you're asking, can this read overlap with any other read, you're asking a really big task of the computer. This read can be overlapped with any of millions of other reads. And you're not done after you've solved that one problem. K-mers represent a very useful indexing strategy for us to resolve that. We're going to create a sorted list of all K-length sequences that could be drawn from any read. All right, so if I have a read of 50 base pairs, I can then say, what are all of the 25-letter sequences, consecutive letters, that can be pulled from this? I've created a K-mer catalog for just that one read. But here I'm going to make a K-mer catalog spanning all of the reads. It sounds like an awful lot of work, but in fact it's quite easy to do that computationally. So from this you can then sort all of the K-mers that come out of this collection. We're pretty good at sorting stuff in computers at this point. We can then exclude the K-mers that we see bazillions of times. Frequently those will represent repetitive DNA. Overlapping reads, if you've got 2 reads that overlap, they're going to share at least one K-mer in common. So what happens if you've got two reads that overlap by 24 nucleotides, but your K-mers are requiring an overlap of 25? They're not registered as overlapping at all. So this is a minute. The larger you set the K-mer, the bigger an overlap that's required before things are called adjacent. The smaller you set the K-mer, the greater the smaller you set the value of K, the greater the chance that two reads are called adjacent when they really aren't. So we have this kind of pressure on both ends of K. Great. So at the bottom, we have a contig. This contig is being built out of the fact that these K-mers of 25 nucleotides all overlap by K minus 1. Does everyone see that? Okay, so we have the top sequence that is the 5' K-mer in the contig. It is okay to transition from the top K-mer to the second K-mer because they overlap at all but one of the nucleotides. And so by using a K-mer catalog like this, it's pretty easy to say, all right, well, these are, given this K-mer, I can jump to this and only this other K-mer in the library. Projecting through that, we get these long contig sequences generated. As I said, we've kind of shifted to an approach where our de Bruijn graphs reflect edges rather than nodes. Edges rather than nodes. So if I started with a sequence like this that was derived from these 7 mers, here we've got a K value set all the way down to 7, the de Bruijn graph tells us that it is okay to go from ATGGAAG to TGGAAGT because they overlap in all but one of the letters. And that edge is something that we want to incorporate between the two. We don't care so much about incorporating each sequence anymore. That's the Hamiltonian approach. Instead, we're going to include every edge as a way to build this larger contig. Okay, now I mentioned that the paired end sequencing approach is going to give us the ability to know which contigs are near each other. Before, you saw the light blue lines with the dark blue patches on either end. This is just another way to visualize the same thing. Here we have these arcs that represent a pair of reads that are known to be on opposite strands of the same insert sequence. So if we can build a contig that represents this region from these overlaps, and build another contig from this region based on those overlaps, we're able to say these two belong close together. Look, we have, in this case, 3 different pieces of DNA that all bridge between these two regions. So this is a well-attested set of contigs that belong together in a scaffold. Great. Now, frequently, when we deal with an assembly, it's a little unclear what we've done. Yes, the computer chewed away and hemmed and hawed, and its little fans just went zzz for several hours. But did we, in fact, come up with something useful? If we have multiple assemblers that we can use on the data, which one did the best job? What we frequently do is to compare numbers called N50s. And I think that a lot of people don't understand the N50s. I want to try to explain that a little more carefully. We have the biggest contig that we generated, and we have the smallest contig that we generated. Generally speaking, you're going to have a larger number of these very small contigs and a relatively small number of big contigs. So one of the things that we do is to sort all of them from big to small, right? So in this case, our largest contig was 2200, our next largest was 1300, 1200, 1100, 650, and then we had some small fry down at the bottom. I used another tool to assemble the same reads, and it gave me a longest contig of 2600, the second largest of 2200, the third largest of 1010, etc. I want to know which of these two assemblies have the largest contiguous regions. One of the ways that we do that then is to stack all of these contigs from biggest to smallest. This is just a stacked bar graph. And then we set a line at half the total. Half the total. So I'm summing all of these contigs together, and I'm now drawing a line at the midpoint of that. In this case, the line, in assembly 1, the line intersects the contig with length 2200. In the other case, it intersects with, with a contig of 1200. So in this case, the number I report as the N50 for assembly 1 is the size of this contig, 2200, or of assembly 2 at 1200. Those values are frequently given as a figure of merit for an assembly, and generally speaking, you're rooting for a larger number to say that this midpoint lands in a larger pool, in a larger contig drawn from that assembly. But I don't want you to see the term N50 and assume it has no value. It's very valuable for you to know that, to understand how fragmentary the assembly is for this species. In general, you will find that a big genome is expressed in what's called the FASTA format. FASTA was one of the early sequence alignment algorithms. This format is so ubiquitous as to be just ludicrous. You find them everywhere. So I wanted to mention that you're going to see files that look like this. It should be pretty plain. We're looking at a text file. So we have a first line that starts with a greater than symbol. That greater than symbol is the hint that you're at the start of a new sequence in this file. And it has some sort of expression after that, followed by a space typically, some sort of white space, that gives the accession number of this particular gene that we're looking at. After that, we see, in this case, PEP, chromosome, GRCH38, gene transcript, etc. This is what Ensemble makes its headers look like for FASTA. However, I would note that there's a large degree of disparity in how well people annotate, how well they describe in their FASTA files what these accessions look like. This happens to be the example from Ensemble. And then we have a bunch of lines of sequence. Again, people who implement FASTA tend to use arbitrary numbers of nucleotides per line. Some will say every line should have exactly 60 characters or less. Some people use 80 instead. It's almost wholly arbitrary. So the first line is the accession and description. Everything else is just sequence. How do you know that you're at the end of the sequence? There are two answers to this. You will know that you've come to the end of the sequence when the next line starts with a greater than symbol, which is to say it's the next sequence. The other possibility is you hit the end of the file. So it's not that intuitive a system, but it helps to know how these are interpreted. It's not like a FASTQ file where there's always going to be a multiple of 4 lines in the file where each set of 4 lines represents a different read. Okay. Lots of pieces from this lecture today. I really appreciate your hanging in there for such a long talk. Sequencing experiments are analyzed through a cascade of bioinformatics steps. We don't start with the sequence data that comes to you from the facility is frequently in just a FASTQ format, and there are a lot of steps to turning that into a product that you can infer biology from. Error propagates at each step. If you trim your sequences or don't trim your sequences, it's going to have a very big impact in what you see downstream. Mapping aligns reads to existing annotation, while assembly builds long sequences from reads, long contigs typically. So I would really be sure you have very clear in your mind why mapping and de novo assembly are very different applications from sequencing data. Collecting contigs into scaffolds is greatly aided when you can make use of paired end sequencing. That is very much a standard affair. If you're going to be doing assembly, you'd better have paired ends. And of course, repetitive sequences can make distant features mistakenly appear close together. They are bedeviling. But we've got a lot of different systems that we use for handling these challenges, and I hope that today you have a better appreciation of just how difficult it is to go from sequencing data to these biological information sets.